I'm sorry that I have so short today, so I'll try to deliver this as clearly as possible. Uh, Today I'm going to talk about the interaction of the immune pathogen on plant in presence or absence of the plant pathogen. So I'm here looking at the interaction of Salmonella and Gautam on the tangle fruit surface. So we know that we had in the past many contamination issues coming from the consumption of the microbes, and the mechanism of the contamination is still unknown. So when we see how the microbe develops, initially you can see here there's nothing on the fruit surface. It's very cute looking, shiny, or sometimes it has hairs on it, but it's very nice to see. But later, at around 10 to 12 days, you can see these kinds of cracks on the fruit surface. And when we minutely observe those cracks, you can still see some lesions coming out. Those are red in color. So later, they heal up like these kinds of lesion. Well, at maturity, when you buy cantaloupes, they are off in surface. So that's actually this is actually the initiation of lating process, and finally you see lating over the tree when they mature. So we don't know the interaction of the salmonella on the cantaloupe fruit. So I'm trying to know about those and trying to get those information. So I have here objective set up saying that whether the salmonella can internalize through those cracks alone or in the presence of the plant pathogen. And the second objective is to understand whether the plant pathogen, presence of plant pathogen can help the salmonella survive on the surface for a longer time or not. So we expect that with the previous reports, the other microflora, they usually help for the feet of the human pathogen on the plant surface. So we conducted research inside the greenhouse, and after flowering, the female flower usually produces the complete flower and female flower. So female flower are generally produced in the later season, and they start with flowering, with showing up the male flower first, and after like 2 weeks, they start with the complete flower. So I was the only person responsible for pollination, so we don't have insects inside. So I was collecting the pollens from the male flower and pollinating the female flower, the complete flower. So after the fruit says, we can see here some cracks. So it is clear here. So you can still see some films or resins coming out from those cracks. And those are the natural cracks. So, so it is how the cantaloupe develops. So these are the wounds you can see clearly here and here. And then those cracks were marked with the 2 by 2 centimeter square. And then 25% of the cells of the pathogen were placed and sprayed with the sterile cross. So I'd like to talk a little bit about the pathogens that we developed. So these pathogens were labeled in our lab, aromatic with the GFP and then salmonella with the TS And this is actually done to get their population on the non-selective media, especially for the plant pathogen, because they don't have selective media found there. So we have to do that with the antibiotic resistant genes, that is the antigen for the Aurelia and then gentamicin for the salmonella. So I would like to go in detail about the treatment, what I did. So the control was with 1%, 0.1% tetron and salmonella alone, or Arunia triticula alone, or both of them together on the spot, but on the different fruit. I had one spot fruit, and only one fruit on the plant was inoculated, and it was allowed one additional fruit to see if the pathogen can move from one fruit to another. So sampling was done right after inoculation, and 9 days after inoculation, and 24 days post inoculation. Here 24 is the average date calculated with the maturity date of the fruit. Like this was sampled at the time of maturity. So a laboratory analysis was done after taking out those fruits from the greenhouse, and those were caught aseptically in the PSO2 layer hood, aseptic cabinet, and the rind and the mesocarp underneath the rind was treated separately, and they were enriched with the universal free solution crop. So for the salmonella detection, initially we started with the trying to know the population on the surface of the rind or on the mesocarp, and there was the excellent that is the selective area for the salmonella because they show up with the black colors. And the other is the enrichment, like 24-hour 24-hour incubation, and then straight to excellent flares again. And then the enriched rod was again transferred to selective area, that is the Rappaport Marceliatis rod, which is selective for salmonella. And then after like 24 to 48 hours of incubation at 42 degrees Celsius, it was transferred to XLE plates or straight to XLE plates to see if salmonella is present there or not. And then the enriched rod was one envelope rod was taken out, and then the centrifuge and the pellets were on PCR to know if we missed those pathogens on the culture media or not, to confirm. So similarly, the Armenia was detected by the same procedure, but we made the NAP plates, mutant plates with the ampicillin amine plates, so selective media for the Armenia, and the other processes were similar. So here we found that we were not able to get a good number of salmonella from the fruit surface after sampling even at day 0. And the population of salmonella went much lower at day 9 and day 24. But still, we were able to find their presence or absence after enrichment, like 24 hours of the incubation. If they are present, then even a single cell would multiply, and then we can see that whether they were still alive or not. But the population count was very low at day 9 and day 24. So again, on the salmonella on the rind surface, in the day 0, we were able to find almost 100% of the samples with the salmonella alone or both of them together. All the fruit samples showed the presence of salmonella. And at day 9, the population of salmonella on both of these inoculated conditions were quite more or less similar, but we found a little bit less, like 2 of the fruit samples were not so positive for the presence of salmonella. And we can see a difference here. With the presence of the Armenia, we have more salmonella arriving in the fruit compared to when it was inoculated on the fruit surface. So let's go towards the mesocarp right underneath the rind. So what we found is none of the mesocarp of 67 fruits inoculated with the salmonella alone or both of them together were negative for the presence of salmonella. We didn't find any salmonella inside the fruit. And the additional fruit on the same plant was also not having any salmonella or any salmonella. So let's talk about the Armenia triticula. What's the condition of the Armenia triticula on those fruit surfaces? Actually, this pathogen is transmitted by there are 2 the beetles, spotted Pokemon beetles, and there's one more. They transmit while they are feeding on those vines. So it causes wilting on the kind of plant. So here we found the recovery of the Aurelia was low even at day 0. We didn't find enough number of Aurelia at the time of right after inoculation, and the difference here we can see is the more error bar in the both of them inoculated conditions. We found that Salmonella was inhibiting the growth of plant pathogen on the culture medium. So we were not able to detect most of the Aurelia on the both of them inoculated condition. And so what we see on the right is the lesions, usually sampled at 9 days and 24 days post inoculation. So here you can see the lesions here also. So it seems like it has gone through these openings that I told you the lesions we have after cracking. So they might have gone through these lesions, so they are able to produce some water-soaked lesions underneath. And when we observe those lesions under the UV light, we found they are still, like they are also multiplying outside, like they are getting nutrients after leakage from the fruit. So they are also multiplying on the surface. So you can see the green patches over here, and still they are also extending beyond the point of evaporation. So maybe they multiply inside and then also multiply surface on the other side of the fruit. So we found that there was not much difference whether plant pathogen or human pathogen and plant pathogen both were inoculated on the water-soaked regions, number of fruits with the water-soaked regions, so there is not much difference. So what we see is at day 9, about 61% of the fruit samples plant pathogen only inoculated lesions, and when both of them were inoculated, we found 56% of the fruit were having lesions. So this is not statistically significant. So I don't think that there is an impact of the human pathogen on the survival of the plant pathogen. So we found that the fewer fruits with the both of them inoculated were there were less lesions on the fruits when both of them were inoculated, but not significant. So on this, so what's there on the subline of the subline mesocarp? That is the right underneath the rind. So we analyzed the mesocarp. So what we found is you can see the water-soaked lesions spreading throughout the whole, almost over the area of the spot. So when we observe these lesions under the UV light, as we have the fluorescence labeled green fluorescence, so what we see is the ammonia was spreading throughout the fruit. So we saw this. But the additional fruits on the same plants were not having any ammonia. So what we found is Salmonella was able to survive on the fruit surface for a longer period of time, still at the time of harvesting. And we don't know about whether Salmonella went inside the fruit or was not able to survive on those tissues. And we now know that with the presence of ammonia, the Salmonella population, Salmonella can survive for longer period of time when both of them are inoculated and with the lesions. If there is no lesion, there is no Salmonella. We found like that. But this was not significant. So it is possible that ammonia directly or indirectly assist the human intelligent pathogens to colonize or survive. So both of these pathogens are developed to the Enterobacteriaceae family. So, and the ammonia can internalize into cameral fluids through the natural cracks formed on fruit surface, suggesting that the opening would allow pathogen entry. And there is no evidence of systemic movement of either pathogens to non-inoculated fruit surface fruit of the same plant, suggesting systemic homeopathism is unlikely. So I would like to acknowledge these persons for directly and indirectly helping for the research, and I would like to especially thank the NINFAB members, the Entomology and Plant Pathology Department, and Major Fund was for the cost. Thank you.